a little bit because I'm not really going to be talking about the elevation gradient so much, but we are going to be talking about POA genetics at various spatial scales. So if you're at all familiar with POA secunda, then you might wonder why we chose to target it for this kind of analysis. I did not cherry pick this picture. This isn't like the perfect picture of POA growing in an agricultural field for seed production that shows how great it is, because this is how you probably experience POA on the ranch, in that you probably don't. You probably experience it when it's under your feet, when you're walking towards something else. So in this picture, we see a really beautiful picture of cheatgrass with some POA underneath it, and you only know that POA is there in this particular case because you can see the seed heads. It's really more apparent when it's in flowers, it has that seed. So believe it or not, this is actually part of the reason why we chose to evaluate POA. So POA's primary role is as an understory interspace species. So that's what it's really good at. It's good at filling in gaps. And as that kind of species, this is a vegetation presence map, basically. So all of these are vegetation survey points that have POA on them. And the most important thing to take away from this is how many of them there are. POA is pretty much ubiquitous across the ranch. And it exists in our grassland sites, like we would expect, but it also is in a lot of other sites that you might not expect, like riparian sites, up at lower and higher elevation sites. So it's able to deal with a lot of ecological conditions. So why is this important? So when we're talking about invasion ecology, open niches are really vulnerable to invasion. Invaders are good at getting into those spots. If they're there, they'll probably get in. So that puts interspace species in the front lines of defense against invasion in a grassland system or in any system where POA is found. So most of you have already seen these figures. These are from a study that Lauren and Dan did. And it has a lot of interesting implications. The data itself, that's really not what we're talking about here. What I'm trying to hit at here is that we see phenotypic diversity in different ranch populations. So these are on the x-axis, POA populations collected across the ranch. And you can see that they vary in their traits. And this is with respect to competition and tolerance of cheatgrass, but it's an indicator that we do see some variation. So what causes that variation? And what we're looking at is whether or not the differences we see in POA across the ranch is genetic, or if something else is going on. And what are the implications of this variation? So you might wonder why go to the trouble of genotyping when we already have some information about how POA acts in terms of its phenotype. So one of the most important overarching reason to do this work is to get a sense of the landscape genetics of native plants. We give a lot of attention to the invaders and what makes them unique and what makes them good at what they do. But it's also really important that we understand native plant dynamics in their uninvaded contexts and how they respond to invasion. So the point here is, if we're going to restore communities, we need to understand how genotypes move through space and time. So how adaptable things are and how they can expand from a given site and how they move through space and time. And how genotypes move through space and time is through reproduction, and that's how phenotype comes from genotype to a certain extent. So, and how disturbances influence patterns of genetic variation. That's also really important as we have sites that have varying levels of disturbance. Also, understanding the genetics of native plants is important for sourcing adaptive variation. In the last few years, the importance of using remnant native plants in restoration efforts has received a lot of attention because they're winning, or they're at least doing well. They're able to persist in these kinds of sites. It's also important for informing plant material selection, which sort of goes back to my last point, and also for the delineation of plant materials transfer zones. So this is sort of we've talked about these earlier, so this is my representation of genetic information and an electropherogram, which we'll talk about a little bit more in a second. So before we get into the genetics work that we're doing here, it's important to take a moment to talk about what makes POA special in some other ways. So when we think about plant genetics, we tend to think about a Mendelian model. We tend to think about Mendel's peas, the outcrossing self-compatible diploid that behaves. And we're not dealing with a species that acts like that. POA has variable reproductive modes. It's self-compatible, so it's got that going for it, but it's also a polyploid. So these pieces of information have a couple of implications, both for how we think about the data we're going to look at today and for how we go about analyzing this species. So POA secunda has 3 reproductive modes, the most common of which is apomixis. So apomixis is asexual reproduction by seed. So this is a little schematic that sort of is trying to get at that. So this is our hypothetical maternal plant, our hypothetical sort of paternal plant, and our offspring. So apomixis in POA secunda is pseudogamous, which means that it requires pollination, but that pollination is not actually associated with any paternal inheritance. Because this is seeding, it's probably just a trigger for the production of seeds. So in this situation, that's what I want. The offspring plant is genetically identical to the maternal plant. That's important when we're talking about how genes move through space and time, because the scale of variation is going to correspond to the scale of dispersal of the apomictic seeds. We don't have pollination happening here, so variation, the scale of variation corresponds with that, with dispersal. It's important to note that this is facultative, so POA is not dependent upon this exclusively. So POA can also self, which is a source of genetic variation in POA. So selfing, just a really quick reminder, is the reorganization of a given pool of genetic information from the parental to the offspring plant. POA also outcrosses. Outcrossing is something we're all pretty familiar with. It's that dual parental model of genetic mixing. So the extent of selfing and outcrossing is largely unknown in POA, and we don't know what exactly triggers the selection of one particular mode of reproduction as opposed to another. Though outcrossing is thought to be quite rare. And again, I'm going to point out that selfing and outcrossing are sources of genetic variation. Our offspring aren't identical to our parental plants, but apomixis is not. It's clonal. So again, patterns of genetic variation are governed by reproductive mode. So the other departure from that model that we were talking about just a moment ago is that POA is a polyploid. So polyploids have multiple copies of their chromosomes. So ploidy in POA secunda is variable. We have a 2N chromosome number that varies between 44 and 106, which is a pretty big range, though we do have modal ploidy at 63 and 84. So why does that matter? This variability in ploidy likely contributes to the plasticity. So we sort of talked about plasticity a little bit when we were talking about the variation in sites that POA can exist at and succeed in. But it also increases the analytical complexity. When you have more genetic information, doing something with that genetic information and understanding what's going on becomes more complicated. In that it also limits our methodological options. So by way of methodological options, we chose to evaluate POA with AFLPs. So AFLPs are a PCR-based method developed by Voss et al. In 1995. So we're using a modified method as described by Blanot et al. 2013. It requires no sequence information, which is fantastic because we don't have any. It's appropriate for polyploid species, but it is a passive marker. So it's a relatively coarse measure of genetic diversity because we're not necessarily showing expressed variation because it doesn't differentiate between coding and non-coding regions. So I'm going to go through very quickly how AFLPs work. So we take our genomic DNA and we put it through our restriction digest. So we take enzymes that are specific to restriction zones in the DNA that break it up into fragments, to which we ligate adapters that correspond with our primers. Those primers are first added in a pre-selected amplification where we take those fragments and we make lots of copies of them, which then goes into a second amplification where we add selective primers. So these are 3 base pair combinations So they only amplify those fragments that start with those 3 base pairs. And those primers are associated with fluorescent labels. So we run samples through capillary electrophoresis, and we get an electropherogram. So these electropherograms are the data. So if you've been around the lab, you've probably seen me sitting in front of one of these with sort of a scrunched up face, kind of a lot. This is by far the most labor-intensive part of the process. So what we do with these is we take replicated samples between and within plates to create bins, which you can sort of see here, which correspond with our alleles. And then we score those bins based on the presence or absence of a peak in them. So that presence-absence data is created by manually scoring the electropherograms, and the output of which is our binary table. So just a word about reproducibility. This is really important with this kind of method. So we evaluate reproducibility, as I mentioned, both within and between plates. We have a 95% bin and sample reproducibility threshold, and we also use positive and negative controls to verify our results. So we evaluated AFLP variation in 5 populations collected across the ranch, as you see here. And those colors are going to mean something in just a minute. We have approximately 20 samples per site that we evaluated. They were collected in the fall of 2012 or the spring of 2013. Green tissue was clipped for DNA extraction, frozen immediately, lyophilized to stabilize, and then extracted with a QIAGEN kit. So that genomic DNA is what goes into the process that I just described. So before we talk about the data, I have to do a little bit of the nitty-gritty sort of gross stuff about reproducibility. Not gross, it's actually really cool, about reproducibility and the extent of polymorphism in our samples. So we, in total, had 80 bins between our 3 primer combinations with an average bin reproducibility of 98.8%, which is pretty good. However, that's really important that it's that high because only about 25% of our bins were polymorphic at a rate higher than our error threshold, which means that about 75% of these bins were identical between our samples. So we're going to talk about what that might mean in just a second. So this is our initial ordination. So this is basically the relationship between samples and ordination space. This is the first and second axis. This is a principal coordinates analysis. We'll look at another axis in just a second, or axis combination in just a second. So before we get into this too much, it's important to note that 16% of our variance was explained by site. So this is a little bit of a messy cloud, but we are explaining 16% of our variance by our site with a p-value of 0.001 with 999 permutations, which is as significant as we can get with this number of permutations. So putting some polygons around this, we can kind of see that there's a lot of, we can see that overlap. We can see the similarity that I was talking about. So that does indicate that there's a lot of similarity between our sites, but that there is some variation. So there's a lot of overlay happening here. But let's look at the variation that's happening within our polygons, within our sites, and try to see what that might mean. So I'm going to use a little bit of site context, because I didn't have a map on the last slide, and I don't know, it's important. So I should mention that the meaning that I was ascribing to these, actually it's probably best to do here. The meaning that I was ascribing to these colors corresponds with the symbols here, which everyone probably already figured out, but just in case. And this is also another way of looking at this in ordination space. So we're looking at the same data, we're just getting a different perspective on how they relate to one another. So, all right. So the first thing I'm going to talk about is the things that kind of look like outliers. It's important to note that this is not the extent of POA sampling on the ranch. We've had some issues with our methods, and we've had getting things above that reproducibility threshold is not a trivial task. So this is the sort of the superstar data that we have to share, but that we have other data that we've added to this, and we're seeing that this pattern of these, so we have these clusters, and then we have some samples that seem to be outliers. As we add samples, we tend to get groups forming around these, which indicates a couple of things. We probably haven't reached the edges of the variation that we see across the range. And then we'll probably start to see things break out as we increase our sample size. So I'm only working with 5 populations here, and that's a limitation. But what we can look at is the patterns that we're seeing in this data. So I'm going to go through the populations briefly and just look at some of the subgroupings that we're seeing. So you may have noticed with our polygon slide that the standout is population 7, which is in yellow, which is, of course, makes it sort of the hardest to see. So we're seeing here and here that we're hitting some pretty clean grouping. There's only one sample that's not in that group in 7. And these are sort of, so this indicates that there's a lot of similarity in the genotype on that site. And then, so moving on to our red, which is our bold cheat site, which is sort of spatially close to that site. We also have one group, but we have a lot of things that are clustering with other groups of samples. And we'll talk about what that might mean in just a second. So moving through this, our green, depending on how you look at it, seems to be clustering into a few groups or a couple of groups, with some samples that are with other groups of samples. So purple, kind of the same deal. And then, so unfortunately, this is the least charismatic color because you can't, it's hard to see. So I don't have an ellipse around this group of samples because they're dispersed throughout all of these sites, which is interesting because this is probably one of the most pristine sites, the least invaded sites, the most diverse sites in this group of samples. We'll talk about what that may mean in just a second. So let's really briefly go back to reproductive modes. So something I neglected to mention in the last slide is that we are seeing some stacking up of samples, which is consistent with what we would expect with an apomictic species. So just a refresher, an apomix is we're going to see clonal reproduction. But that's not all that's happening here. You can see that there's a lot of variation that we're showing, which is probably consistent with some selfing that's happening, which indicates that there's more selfing happening than we kind of thought going in. Because we're seeing some restructuring, but not radical departures, or real sort of intermediate relationships. So that gets back to a question that we haven't really answered here that is really interesting for the future in fleshing out this data set. What are the spatial patterns of variation in a species with variable reproductive modes? So, and to our other slightly strange thing about this species, we talked about how ubiquitous this is across the ranch, which is an indicator that it can tolerate a lot of environmental conditions. So it's also important to note that there's a lot of genotypic overlap between sites with very different environmental conditions. Not entirely, and there's some variation. We have some subgroups, which indicates that a mixture of genotypes can persist in a site, but there's a mode genotype, typically probably that's most successful at that site. There might be a couple of them, and that's kind of what we're seeing with some of our sites. And that's not, I jumped the gun, sorry. There's the mode genotypes that may be most successful at that site. So in conclusion, we have shown that there is genetic variation in POA secunda across the landscape, but this variation is not entirely random. We're seeing significant correlation with site differences. We've not yet established the scale of that variation, and there are signs that selfing may be occurring with more frequency than we anticipated. There's also some support for the importance of phenotypic plasticity in the species, which isn't exactly new information, but it's always good to sort of come back to those kinds of core understandings of how a species works. So by way of future directions, we aim to increase our sample size, to increase spatial resolution and capture more of our variation, to explore relationships between genotypes and phenotypes, genotypes and environmental variables, and space, that elevation gradient we were talking about before, well, that I didn't talk about. And also to determine the ploidy of some of the ranch populations to see if that co-varies or relates to how these guys behave in terms of their landscape genetics. So, questions?